Hello friends, I am Dr. Shital Thacker. I am Assistant Professor at Rai School of Science, Rai University, Ahmedabad. Today we are going to discuss about very important and very interesting topic of biology. So, we are going to discuss about the structure of protein. But before we start, I would like to give you Ande ka funda. So, we all know that eggs is one of the delicious breakfast in all over the world. But when we fry the eggs becomes white. So why eggs becomes white? Have you think about that? Yes. So let me explain. Egg is made up of lots of proteins and one of that protein is albumin. Albumin has made up of lots of amino acids. So some amino acids are hydrophobic. Hydrophobic means the amino acid which repel the water, which gets away from the water. So these amino acids, when we fry the egg, it will get away from the water and it will bind each other. So it will form a network and this network will convert it to the white color. So this process is the protein denaturation process, and due to the protein denaturation process the egg becomes white and we can have the delicious omelette in our breakfast. So thank you, protein denaturation process. Before we start the structure of the protein, let me explain something about the protein. We can find protein in various form. We can find protein in muscles, we can find protein in the bones, we can find protein in hairs. In each and every tissue is made up of protein. Enzymes are made up of proteins which will help us in the infection, to fight the infection. Even the hemoglobin, which carries the oxygen in our body, is also made up of protein. So proteins are larger biomolecules and macromolecules that is made up of long chain of amino acids. So before we start the structure, I will give a brief introduction about the history of the protein. So protein is a Greek word. Greek word is proteos. This proteos word is given by two scientists. One is Mulder and Berzelius. These two scientists gave this word, and this word means primary, which is most important. So protein is one of the most important biomolecule of life. And protein, the first protein which is whole sequenced is insulin, and it was done by Frederick Sanger. And in 1958 Frederick Sanger received Nobel Prize for the sequencing of insulin. This is the structure of the insulin. The insulin is made up of two polypeptide chain. One is A chain and second one is the B chain. This two chain is interlinked with disulfide bonds. So due to the first sequencing, we know the working and structure of the insulin, which was given by the Frederick Sanger. Now, as I said, that protein is made up of amino acids. So the monomer of protein is amino acid, and amino acid is polymer of amino acid is protein. So when there is one, it is called amino acid. When there is two amino acid, it will be called as a dipeptide. When there is 3 to 20 amino acid is interlinked with each other, it will be called as an oligopeptide. And more than 20 amino acid is interlinked, it will be called as a polypeptide protein. So amino acid, we all know that amino acid, it says that there is the amino group and there is a carboxyl group of acids. So amino acid is made up of two groups. One is amino and second one is the carboxyl. So let me just explain the basic structure of the amino acid. Amino acids have four covalent bonds. One bond, which is this is our alpha carbon. So first, this is the main carbon, which is called as alpha carbon. This alpha carbon is binded with the carboxyl group with covalent bond. The second bond is with amino group. The third bond is with hydrogen group, and the fourth group is with R group, or we can say it is the side chain. So every amino acid has same carbon group, same alpha carbon group, same hydrogen group, same amino group, same carboxyl group. Only different amino acids. We have 20 different amino acids. So these 20 different acids differs only from the R group only. So this is the structure of the amino acid. This is amino group, this is carboxyl group, and this is hydrogen. So in all amino acid, these three groups are same. Only the difference is of this side chain, which is R groups. So 20 different R groups made 20 different amino acids. So now we will look at how protein synthesis is occurred. So polymerization of the amino acid will prepare our protein. So when two different amino acid will react with each other and it will form a peptide bond. So the carboxyl group of the one amino acid, the amino group of the second amino acid, they will react with each other and form peptide bond. This reaction is called dehydration reaction, or we can say condensation reaction. This reaction will form the peptide bond and we form the protein. When this peptide bond is break down, this process is called hydrolysis process. So this is how 1, 2, 3, 4 multiple amino acids will form a peptide bond and we can prepare the proteins. So initially when there is no analysis technique was there, right now we have lots of analysis techniques. We can confer the structure of protein at the atom level, but before that, when there is no analysis techniques, biochemists made the 4 different levels of the protein to make the ease to understand the structure of the protein. So there are 4 levels. First is the primary structure of the protein, secondary structure of the protein, tertiary structure of the protein, and quaternary structure of the protein. I will brief everyone about this 4 level in this PPT. So the primary structure of the protein is a linear sequence of amino acids. As I said, there are more than 20 proteins are there. So in each protein have their specific sequence of amino acid. In every polypeptide chain, there is always first amino acid has a free amino group and always last amino acid has free carboxyl group. And between these all amino acids, there is peptide bond. So the first amino acid's carboxyl group and the second amino acid's amino group will bind each other and form the peptide bond. And this is the sequence of any one amino acid. Each amino acid has specific sequence. If there is a change of only one amino acid, it will change the structure of the protein and it will also affect the function of the protein. Now we will see the secondary structure of the protein. So in the secondary structure of the protein, there are basically 2 secondary structure. One is alpha helix and second one is a beta plate sheet. So in secondary amino acid, there are hydrogen bonding is seen between the amino acid's carboxyl group and hydrogen of the amino group. So in one amino acid's carboxyl group will form the hydrogen bonding with the second amino acid's amino group. In beta plate also there is hydrogen bonding. Now how alpha helix and beta plate differs with each other? In alpha helix, one amino acid is far away with the fourth amino acid and it will form the hydrogen bonding and it will make a turn, right-handed turn, and there is 3.6 amino acid far. So in one turn there is 3.6 amino acid and it will form right-handed turn type structure. That's why it is called alpha helix. In beta plated sheet, the sheet type structure is made up of, and there is compulsory to have 2 sheets to make the hydrogen bonding between beta plated sheets. So in one polypeptide chain is connected to the second polypeptide chain, and this could be the anti-parallel also and parallel also. It will form a plate-like structure also. It could be form a zigzag type structure also. So in the secondary structure, mainly hydrogen bonding is found, and this bonding will not be occurred between the R group of the amino acid. Third is the tertiary structure. In tertiary structure, the further folding occurs in the secondary group. So we know that in secondary structure there are various R groups. So there are basic R group, hydrophilic amino acid, hydrophobic amino acid, acidic amino acid, and sulfadic amino acids. So the different bonding, ionic bonding, polar forces, and covalent bonding, disulfide bridges will occur with each other, and in this bonding R group is involved. So the different bondings, weak bondings as well as the strong bondings like van der Waals bonding, ionic bonding, electrostatic bonding happens between the R group of the amino acid after the secondary structure, and this will form the 3D structure of the amino acid. So when the denaturation of the protein occurs, the 3D structure gets degraded, and due to that the function of that proteins hampers. Last is the quaternary structure of the protein. So quaternary structures only occurs in the protein which has multi-subunit protein. If the protein is made up of only one polypeptide chain, it will not form a quaternary structure. There should be more than one polypeptide chain to make the quaternary structure. So in the quaternary structure also R group is involved. So this is one polypeptide chain, this is second polypeptide chain. In one polypeptide chain, this is the 3D structure of the tertiary structure of the one chain, and this is 3D structure of the second chain. So between two subunits there are different type of bonding we can found. This bonding could be the disulfide bonding, this could be bonding the van der Waals bonding, hydrogen bonding, ionic bonding. Any type of bonding between two subunits can form a quaternary structure. We can see the hemoglobin which has 4 subunits, and it will form a quaternary structure to carry the oxygen and to transport the oxygen to our body. So we have seen there are 4 levels of the protein: primary structure, secondary structure, tertiary structure, and quaternary structure. In primary structure, sequence of the amino acid is there, and peptide bond between the amino acid was found. In secondary structure, folding occurs in the primary structure, and the hydrogen bonding is found between the same polypeptide chain. In tertiary structure, folding continues. Covalent bonding, hydrogen bonding, van der Waals bonding, and different other bondings will found between the polypeptide chain, and in this type of bonding R group will be involved. And last but not the least, quaternary structure, where folding continues and different types of bonding will occur between different type of polypeptide chain, and it will form the quaternary structure. So in this three structure, only one polypeptide chain or one molecule will be found, and in the quaternary structure we can found the several molecules, and this is how the protein is made up. So this is the basic structure of the protein, and I'm completing my lecture now. If you have any query, please comment in your comment box. Thank you so much for the listening.